Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is visible. Please let me know in the chats if I am audible and visible. Yes. Are we live yet? Yes, we are live, right? Awesome. So today we are going to do neural control and coordination. For those students who do not know, this is one of the most important chapters of your human physiology, and if you're learning it for the first time, then you're in the right place because I'll be teaching you from basics. Like, I will just treat you as a student who doesn't know anything. So this way what happens is we'll be able to build it up, right? Awesome. All of you, if you're new to the channel, quickly smash the like button and also mark your attendance, because this way we'll be able to know who are the students who are with us in the live. Okay, so students, neural control and coordination has many topics. We are going to divide this class into two parts. First topic, we are going to learn about all the types of neurons, how an action potential is created, okay, and then we will go into the rest. Okay, hello students, hi, welcome you all, good afternoon. Okay, we reached good afternoon, I guess. Yes, good afternoon. Okay, awesome to see you all. And the chat box was lit. No, the chat box was lit. Very happy for that. Yes, so students, then see, the marks weightage is around 3 to 4 questions. That means you cannot even think of skipping this chapter, because this chapter gives you 12 to 16 marks. Okay, 12 to 16 marks is no joke, because you know every single mark is important. Okay, every single mark is important. Okay, my health is getting better. Sometime back it got a little more worse. Then again, okay, it's little more better. It's going up and down because here Bangalore weather has changed drastically. It's raining in the morning, then it is sunny in the afternoon, then again it's raining, then it's hot. So because the weather is changing, even our body is not able to cope up with it. Okay, so yes. Chalo, shall we start, students? So now every thought process you have, right? Everything that you're thinking, everything you're doing, everything is in a coordination. Yes or no? You have thought that morning I have to attend Gopika Ma'am's class. You made the day like that. So you're working in coordination. Same way our body also works in coordination with mainly two people. Okay? Students, it is RNA. I am going to write the notes. So better let the energy be good and let the likes be good, and also subscribe to the channel. Okay? Because you people want me to do more RNA classes. I am taking more RNA classes than NCERT line by line. Okay, so yes, see students, so everything happens in a coordination. Yes or no? You do everything in a coordination. Now from where does this coordination come? Coordination comes because of mainly two systems in our body. Those two systems are nervous system, which you are going to study today. Okay, nervous system, and second one is going to be your endocrine system. What is it? Endocrine system. Endocrine system. Now you can have a question, no? Ma'am, nervous system also is doing the same thing. Endocrine system also is doing the same thing. Are they actually doing the same thing? Not actually. Nervous system has the ability to get excited. Now imagine we have Meghana in the class, or we have Abhi in our class, Dinesh in our class, Hasini in our class, Savita in our class. All of them are able to react. Right, they're able to respond to a stimulus. That means that excitability is coming because of the nervous system. So these people have the ability to get excited, right? They have the ability to get excited. So excitability is there. Excitability is there. And you people tell me, how does a signal go in a nervous system? All of you in the chat box. Yes. Okay, come on, tell me, how does a nervous system work? Ma'am, by impulse, ma'am. Impulse is coming. They are transmitting that impulse and it is going further. Yes or no? Agreed, all of you? Yes or no? So here it works on electrical impulse, right? It works on impulses. That is, action potential will be created when a stimulus comes, and then depolarization happens, repolarization happens, this continues. So I would say that an electrical impulse is happening. Yes or no? An electrical impulse is happening. All of you agree or not? But at the same time, if I have to talk about the endocrine system. Endocrine system, I know it is a gland. And here what happens, students? Blood is doing the transport. Right? Blood is doing the transport with the help. Because here they do not have ducts, right? They do not have ducts. They are ductless glands. Okay, Ductless glands, exactly. Here ducts are not there, blood is doing the job of transport. Now all of you in the chat box tell me, will nervous system work faster or endocrine system will work faster? All of you in the chat, quickly tell me the answer. Let's see who is the fastest finger. Okay, fastest finger will win. What you will win, I don't know, but you will win. Okay, so fastest finger will win. Let me know in the chats, nervous system according to you will work faster or your endocrine system will work faster. Quickly. Quickly. Yes, Sindhuja has answered first, then we have Abhi. Okay, very nice. Okay, some are telling ma'am endocrine will work, some are telling nervous system will work. Now students, remember, if an impulse is going, you're sending a message through WhatsApp, the message will be reached the moment you type and send. Right, that person will receive the message. But at the same time, imagine I'm using a mediator to transport the message. I'm using a postman, I'm using a bird to transport the message. It will take some more time. But the catch here is nervous system works faster. Okay, nervous system works faster, but it is not long lasting. But it is not long lasting. But imagine I'm sending a letter, right? WhatsApp messages, immediately I'll delete it. But imagine I'm sending a letter. The proof will always be there with that person, right? So this is compared to nervous system slow, okay, slow, but it will last for a longer time. Is this clear to all of you? So these two people are going to do the entire coordination of your body. If you're working properly, that is it. Okay? No, Tanuja is also there. Sindhuja is also there in the chat box. Okay? Got it? Tanuja, Sindhuja also is there in the chat box. Okay? She's my DNA student. So yeah. Look here students, these two people are going to do all the set of activities for your coordination to happen. Is this clear? Okay? Coordination to happen. So the most important thing is this fellow is fast. Who? Neural system is faster. This fellow is slow. Who? Endocrine is slow. But this fellow is not long lasting. But here, this is more lasting. Obviously, because here, hormones are getting passed, right? Hormones are getting passed. So, you have to remember that. Very, very important. Students, next chapter is chemical coordination. No? That time you will study about your endocrine system. Introduction clear? Okay. Introduction clear? Awesome. Now, when I talk about nervous system, right? Okay, when I talk about nervous system, tell me what is the first thought you have. Like, ma'am, when I'm talking about nervous system, okay? Okay, yes. Yeah, my voice is changed because of the cold. Okay, because of the cold, my voice is changed. Never mind. Is the target reaching, students? Target is reaching? Not at all. Students, I want all of you to mark the attendance. Then only I'm going to proceed in the class. This is not fair, right? I'm standing here and I'm teaching you people. All you have to do is mark your attendance. Mark your attendance right now. I've always told you this. This is the little respect that you give for the teacher. That, ma'am, I have come to the class. I will mark the attendance and only leave, right? You shouldn't leave also. You should stay till the end. All of you, quickly. I want the likes to increase. Faster. Which chapter is happening? Who is asking that? All sleepy, wakes up in the morning and opens YouTube. Okay, whichever chapter is happening, I'll do the chapter. Quickly, I can see everyone who marked the attendance. Come on, put thumbs up in the chat. I can know the numbers, okay? So don't lie. Okay, chalo. Very good. Okay, now students, look here. Whenever I talk about nervous system, the first ever thought that comes to me is neuron. Yes or no? Students, many students get confused between neuron and nephron. Please don't get confused. Here you are talking about neuron. What is this neuron? This neuron is the structural, okay, structural and functional, okay, structural and functional unit of who? Of nervous system. Of nervous system. Yes, yes. Okay, JEE students also are here. So you will not see, now attendance came. Okay, one dose of scolding is needed, then only they'll do it. So look here, students, it is not just like, okay, ma'am is asking likes. It's not like that. See, when you like, it reaches to more students, and I'll be able to reach more students. Okay, there are a lot of students who struggles with like understanding small concept. Maybe my way of teaching or putting some examples, putting some joke might help a lot of students. So I want to reach to more students, and that can only be possible because of you people. Okay, how much ever good I teach, how much ever I stand here, even if I'm sick, even if I have headache, how much ever I stand here, no use until you people do the effort. Okay, so that's why it is important. So make it a habit. Okay, chalo. Okay. Yes, structural organization will do it. So here what is going to happen, it is the structural and functional unit of nervous system, means all the job that you are doing, right, all the job this nervous system is doing is because of a neuron. Okay, all the job this nervous system is doing is because of a neuron. Is this clear to all of you? Chalo, very good. Okay, now students, in nervous system, actually when I teach you structural organization, maybe we'll talk about this, but otherwise we have two main cells. Okay, two main cells in our nervous system. Okay, I'm writing it on this brain only. Okay, two main cells on the nervous system. First is your neuron. All of you know about neuron, no doubt in that. Okay, neuron. Next is neuroglial cells. Okay, neuroglial cells. Now, nervous system or your neuron has the ability to get excited. Yes, ma'am, no doubt in that. Whenever a stimulus is there, this fellow will get excited. But what about this neuroglial cells, students? This neuroglial cells cannot get excited. They don't have excitement. They are like this, you know, people who sit in the class, no, how much ever we tell them, if you're dancing also, they'll be like this, they'll sit, no, not excited they are. So, this is excitable. Okay, excitability will be there. This is non-excitable. Excitable. Then you can ask me a question. Ma'am, this fellow is the structural and functional unit. Okay? Structural and functional unit. This is not the structural and functional unit. Then why are you teaching us? Why are you teaching us, right? This is not doing, this is not getting excited, nor it is the structural and functional unit. But neuroglial cells constitute around 50% of the volume of your nervous system. Is this clear to all of you? Neuroglial cells are very, very important. Why? Because they are going to do the job of packaging, okay, support and packaging for the system. Okay, support and packaging of your neuron. Support and packaging means what? If I'm packing something means what? I'm giving a gift box, right? In that I'm packed the gift box. That means I'm putting a cover. Okay, I'm putting a cover on it and I'm giving it to someone. Telling that, okay, take it. Okay, that means I've put a cover. Same way, this neuroglial cells are going to do the support and packaging of your neuron. That means what? Maybe some cells are there or myelin sheath is there. All this, can you all relate? Okay, can you all relate? Yes, you cannot remember each other, but you can remember these topics. Come on, all of you. Okay, so students, neuroglial cells constitute around 50% of the volume of your neuron, and these two cells are very, very important. In that, neuroglial cells does the job of support and packaging. Clear to all of you? Is this clear? Chalo, let's move to the next topic. Okay, Now students, we have to understand what is this neuron constituting, how does this neuron look like, what are the types of neuron. Okay, I think if the types are not there, let's duplicate the slide. Okay, here we have a slide. Okay, ready to draw neuron with me, all of you? Last time also we drew, this time also we're going to draw. Okay. Draw the neuron with me, all of you. 1, 2, 3. Okay. 1, 2, 3, 4, 5. Yeah. What am I drawing, students? This is dendrites. All of you have done this in your smaller classes. You don't need much explanation for it. Oh no. Okay, I'm drawing the, okay, axon is done. Okay, axon is done. 1 second. Students, all these notes you will be available on Vedantu NEET English Telegram channel. So quickly go and join the Telegram channel. And if you want to join my Telegram, it's Biology by Gopika. Okay. This is a neuron. Okay, this is a neuron. Now let's complete the diagram. Let's complete the diagram. In middle there is going to be a nucleus. Obviously there is going to be a nucleus, and inside that there are going to be cells. Okay, cytoplasm will be there. Inside the cytoplasm, so many organelles will be there. Your SCR will be there. RR, rough endoplasmic reticulum will be there. All this will be there. Okay, down when I'm coming, I'm going to see an axon. Okay, so this is the dendrite, this is the axon, and this is the axon terminal. Before that, let's label the main portions. Students, this whole portion, right, this whole portion is going to form my cell body. It's going to form what? Cell body, or your cyton. Cyton. You can all see, no, if I write it here. Cyton. Okay. Now, this portion, right, it's not exact separation, but this portion is going to be my axon. This portion is going to be my axon terminal. Axon terminal. Okay, ma'am, understood. Okay, ma'am, understood this. Okay, this is your cyton or the cell body, and this is your axon, this is your axon terminal. Now let's label each part. Okay, let's label each part by part. So students, this one which you're seeing, no, this extensions, these are called your dendrites. I don't think any student will have doubt in this, right? Dendrites, you have studied in your smaller classes as well. From the dendrites, you can see from the cell body, this dendrites emerged. This one is going to be the cell body, this one. Okay, inside one I'll label it as cytoplasm. Okay, cytoplasm. Inside this, the middle portion, right, you can see, you know, this is your nucleus. Since inside the cytoplasm there will be many organelles. Okay, many organelles will be there. Rough endoplasmic reticulum. Done. Nucleus is there, cell body is there. Cell body has different terms like your cyton, soma, that we will write it separately. From there down, if I come, students, they have a specialized cells called as Schwann cells. What are those cells called as? Schwann cells. This one. Okay, these cells. These cells. These cells will release. These cells will release a white color sheath called as myelin sheath. What we used to do in smaller classes, when we never used to label Schwann cells, we used to write that myelin sheath because it was too difficult to learn it. Now we know that these cells are present, which are called as Schwann cells, that release myelin sheath. Okay, got it all of you? Chalo. Now look here, I'm continuing the drawing. Okay, again I'm drawing some Schwann cells. Okay, cells are done. Because my axon is very straight, the cells also are very straight. Okay, so here you can see Schwann cells are there. So let's label that as well. Schwann cells that release myelin. So this one which I have drawn is a myelinated axon. Yes or no? Because myelin sheath is there. Okay, because these people will release a white color sheath called as the myelin sheath. Okay, now as I come down, students, this is the axon, right? Long axon is there, no need to label it. Between this, between the myelin sheath, can you see a spot where the axon is getting, you know, axon is getting exposed? Okay, axon is getting exposed. That portion is called as your node of Ranvier. Okay, node of Ranvier. I know you will never forget this because it's one of the actor's name. Okay, node of Ranvier. Okay, but the spelling is different. Node of Ranvier is the portion where the axon is myelin sheath is absent and the axon is exposed. Okay, here what is happening? The axon will be exposed. Axon is exposed. Okay, ma'am, understood. As I come down, I will be able to see what? Here there are axon terminals, which I can call it as synaptic bulb. Okay, synaptic bulb. Students, the bulged portion, no, the bulb-like structure is synaptic bulb, or even you can use the word axon ending. Okay, I have written there, no, axon terminal. You can also write synaptic bulb. Now, okay, ma'am, understood. Now, students, this, if you go back to your cell chapter, all of you know cell chapter, no? Baswa sir had taught you cell chapter, yes or no? Yes, quickly. Yes, tell me the function. Tell me the function. Quickly. What have I written? Synaptic cleft is there, synaptic bulb you can tell. Synaptic cleft is the between portion, that when we draw the axon you will understand. Okay, don't take tension now. You can call this as the synaptic bulb. All of you tell me what's the function of endoplasmic reticulum. Okay, endoplasmic reticulum. They can form proteins. Okay, this rough endoplasmic reticulum, they can form proteins, yes or no? Okay, yes or no? Yes. Those protein, students, they are called as Nissel's granule. Okay, they are called as Nissel's granule. So look here, I am drawing some green color dots, if you can all see. These are the granules, Nissel's granule. See, can you see these dots? Okay, these are called as Nissel's granule. Nissel's granules. These Nissel's granules have the ability to form some neurotransmitters. Very good. Protein synthesis. Exactly. So these are your RERs, that is rough endoplasmic reticulum. Okay. They can form some protein, right? They can do protein synthesis. This you have studied in your cell chapter as well. Okay. They can form, or they can do protein synthesis, and they will form a set or one type of neurotransmitter. Okay. They can form one type of neurotransmitter. Only one type, okay, not all of them, one type. One type of neurotransmitter. And where these neurotransmitters will be stored? The neurotransmitters will be stored in the synaptic bulb. Is this clear to all of you? These granules are called Nissel's granules, which are present in the cytoplasm. Okay, cytoplasm of your cell body. Okay, cytoplasm of your cell body, and cell body or soma or pericardion. Okay, they, as you come down, you see them, they will emerge into the dendrite. They will not come extend to the axon. This Nissel's granule, no, they'll emerge into the dendrites. They will not come towards the axon. This you have to keep in mind. Now, look here, students. The axon, no, has two portions. Two portions you have to remember. The plasma membrane of the axon is called as axoplasma. Okay, the plasma membrane of the axon is called as axoplasma. Axoplasma. Okay, axoplasma. Clear? Now, let's go a little more. The plasma membrane of the axon is called axoplasma. What will be the membrane called as? Membrane called as? Think and tell me, all of you in the chat. Yes. Sorry, cytoplasm of the axon is called as axoplasma. Okay, I'll write it here, cytoplasm. Okay, otherwise we'll get confused. Cytoplasm of the axon is called as axoplasma, and the plasma membrane will be called as axolemma. Okay, axolemma. Ulta, Rani Ulta. Okay, this is axoplasm, that is the cytoplasm. Axolemma is the membrane. Okay, yes, very good. Most of you did it right. Okay, this membrane, this white color which I have drawn, no? There's no place to write it here. Where do I write it? Okay, see here, this is called as the axolemma. Axolemma. Here inside this is your synaptic bulb. Okay, now students, this neurotransmitter is going to be present inside your synaptic bulb. Okay, this neurotransmitter is going to be present inside your synaptic bulb. So if I draw an enlarged structure, how will it look like? Yes, quickly in the chat. Okay, inside the synaptic bulb you will have a synaptic vesicle. So I'm going to draw it. I'm taking an enlarged structure of this. Okay, enlarged structure of this. See, look here. Oh, black. Okay, students, look here. This is your synaptic bulb. Okay, I'm just drawing in 3D type of image. This one, inside that, let's say with orange. Okay, this is your synaptic vesicle. Okay, inside the vesicle, who is going to be there? Neurotransmitters is going to be there. Okay, inside the, who is going to be there? Say acetylcholine is there, something is there. Okay, someone is there. Epinephrine is, someone is there inside this. So this becomes your neurotransmitter. So the synaptic bulb, inside the synaptic bulb you have the synaptic cleft. Okay, synaptic vesicle. Okay, synaptic vesicle. Vesicles. Synaptic vesicles. Got it? Done? Write with black color. Wow. Okay, got it? Okay, chalo. So synaptic bulb, synaptic vesicle. Okay, I'm writing that also here. Synaptic bulb. Students, any doubt? Any doubt in this diagram? Please let me know in the chat. Any doubt you have? Okay, Schwann cells will release your myelinated sheath. So obviously here, okay, here I'll write it here. Here, myelinated. Okay, myelinated. Done. Done. Got it? Any doubts so far? Quickly tell me. Okay. Now myelinated, non-myelinated we'll study. That will come under types of neuron. No doubts, right? Thumbs up in the chat, all of you. Thumbs up in the chat, all of you. Chalo. Okay. Done. Now students, as I told you, Schwann cells are there. They will release myelinated. So two types of neurons you can see. Okay, two types of neurons you can see. Let's divide this board into two parts. I have to teach you some more things. Chalo, let that neuron be there. I'm writing here. Okay, neuron is there. Two types will be there. Myelinated and non-myelinated. Okay, myelinated and non-myelinated. See. Okay, this is one neuron. This is one neuron. Okay, here see what I'm drawing. What am I drawing? And whatever you want, you draw. Okay, whatever you want, you draw. Got it. Okay. So here, what do you see? Here you are seeing, no, the cells. The cells that you're seeing, those are the Schwann cells which are releasing your myelin. So this becomes your myelinated neuron. Okay, myelinated neuron, and this becomes your non-myelinated neuron. Okay, non-myelinated neuron. Clear? That means here there are no Schwann cells to release anything. Okay, in this tip, students, your dendrite and axon terminal will be there. If you want me to draw that, I'll draw that. Okay, this is your axon ending. Okay, this is your axon ending. Same thing I'm drawing here. Okay, done. This is your axon ending. So here this is a non-myelinated neuron. That means they don't have Schwann cells and they do not release any myelin sheath. Now what is the job of the myelin sheath? The job of the myelin sheath is very simple, that it has to act as an insulator. Okay, it has to act as an insulator. So myelin sheath's main job, okay, myelin sheath's function is to act as an insulator. Okay, as an insulator. Done. Okay, now see, understand, we have two types of nervous system. Okay, this nervous system I'll teach you. Okay, wait, yes, students, two types of nervous system. We have the CNS and the PNS, central nervous system, and the PNS. Okay, peripheral nervous system. So let's write the word nervous system. Okay, nervous system. We have CNS and we have PNS. Now, both of them, in the CNS and the PNS, okay, they have different people to release the myelin sheath. Ma'am, what did you just say? Right? See, I'll tell you. Students, in the CNS, the myelin sheath will be secreted by or given out, like made by the oligodendrocytes. Oligodendrocytes. Okay? These cells will be giving out your myelin sheath. Is this clear to all of you? Very nice. Madhumati is giving a check. Okay? Oligodendrocytes are the myelinating sheaths. Okay? Myelinating cells in the CNS, that is in the central nervous system. Okay, and they have a lot of myelinated neuron as well. But in the PNS, okay, in the PNS, Schwann cells are the ones that are releasing your myelin sheath. Got it? Schwann cells are the ones forming your myelin sheath. This you should know. This you should know. Okay, this you should know to understand who is doing what. Yes, very good. Very good, Bava. Yes. So, the parts of the PNS and the CNS, we will do it later. But you, all of you know, I hope, CNS has your brain and your spinal cord. Okay, it has your brain and spinal cord. And PNS has what, students? Your cranial nerves and spinal nerves. Cranial nerves and spinal nerves. These are the two things that are present in them. They have a lot of nerves, right? They have a lot of nerves, and oligodendrocytes are the myelinating cells. Done, done, done. Is this clear? Okay, two types of neurons you study. Now more types of neurons you're going to study. Okay, more types of neurons you're going to study. Students, so far any doubt? So far any doubt in the chat box? Let me know. Any doubts? Any doubts? Be it anything that I started from the starting. Any doubts? Tell me. If no doubts, I'm moving forward. Okay. Chalo. Now, different types of neurons are there, may be present at the embryonic stage, may be present in different parts of our body. Okay, different types of neurons are there. So you have to study 4 types of neuron. In that, first neuron is going to be called as your unipolar neuron. What is it going to be called as? Unipolar neuron. Now from the word itself, what do you understand, students? Uni. Okay, uni means single, right? Uni means single. So, uni means single. Polar means to one direction. So, unidirectional I can tell, or single direction. Okay, unidirectional or single direction. That means to one direction. So, let's say if I draw a neuron. Okay, if I draw a neuron. This becomes your unipolar neuron. Okay, yes, this becomes your unipolar neuron. That means only one thing is there. See students, normally what should happen? Let's consider my hand as the neuron. Okay, this is my dendrites. The fingers are my dendrites. Okay, dendrites is there. Let's consider this portion as my axon. Okay, normally a neuron should have what? One dendrite should be there, one axon should be there, or multiple dendrites should be there. Okay. But here what has happened, here this portion, dendrite portion I am folding, and only axon is there. This becomes unipolar. That means only in one polarity, only in one direction, one thing is there. It could be an axon or it could be a dendrite, but here unipolar means one axon is only present. Is this clear to all of you? Okay, only one axon is present. Only one axon is present, and this is seen mostly in the embryonic stage. Okay, this is mostly seen in the embryonic stage. Definitely later development will happen, but this is seen, okay, this is seen in the embryonic stage. Embryonic stage. Okay, embryonic stage. Clear? Yes, present or found in the embryonic stage. This is the first thing. Okay, ma'am, understood. First type you understood. Second type. Second type, you can, from the name itself, you will obviously know it is bipolar. Okay, bipolar neuron. Bipolar neuron. Bipolar neuron means what? Bi means two. Okay, bi means two. That means in two polarities something is there. Normally my neuron should have been like this, right? Now they're telling two polarities neuron is there. That means what? See, look at the diagram, you will understand looking at the diagram. Students, it might look very funny, but I will try to draw it. Usually how do we draw? We draw many dendrites, yes or no? But here, look here, only one dendron and one axon, okay? One dendron, one axon. This looks like a fish that is going to find something, but this is actually a neuron, okay? This one which I have drawn small is your dendrite, okay? Since I am drawing it, you have to agree to it. Is this clear? Clear to all of you? That means two polar region, two extensions are there. One axon, one dendrite. One axon, one dendrite, not multiple dendrites, okay? Normally we draw multiple dendrites. So, one dendrite, one axon. Any doubt? Any doubt? Okay. Now, where is it found? This is found in the retina of the eye, and it is also found in the olfactory epithelium. Okay. It's found in the retina of the eye and also in the olfactory epithelium. Oof. Clear? Only few of them are answering. What happened to other people? Okay, done? Clear, ma'am. No doubts in this. Okay. Next one is going to be pseudo-unipolar. Okay. Pseudo-unipolar. From the name itself, pseudo, false. Right? Pseudo means false. What this fellow did, no students? Okay. False. What he did? Very chalak, okay, very chalak. Starting, he looked like this, okay. We all thought, okay, he's going to form a unipolar, okay. He went all the way like this, There'll be some people, no, in the starting full energy. Looking at the energy, we'll feel like, okay, this year to pakka the student is going to achieve something. In the middle of the year, they will lose all the interest. We'll never even see them in the life. Okay, we'll never even see them anywhere, live, comments, nothing. That is fake, like in the sense, fake promises they give. Same way, people in your, like some friends, some relatives, you'll think that, okay, they are supporting, okay, they're very positive. Later, they will change, okay, complete change they'll do. So, those are like this. Okay, those are like this. What happened is, when they started off, they looked like a unipolar, okay, don't comment on my fish, okay, you have no rights to comment, it's all a neuron. Students, starting, it gave an impression that, okay, it's going to become a unipolar. You saw that? It became like this. After going at the tip, what did it do? It emerged into one axon and one dendrite. It emerged into one axon and one dendrite. And can you see how it emerged? It did not emerge like them. It did not emerge like how it has to be. It emerged in one extension, in the one lane itself it emerged. Got it? So, this is called a pseudo-unipolar. Pseudo-unipolar. Can anyone tell me where do we find pseudo-unipolar? Quickly in the chat box. Root ganglion of the, very good. I think Paul has already told. Very good. Okay. Where do we find it? At the dorsal root or root ganglion of the spinal cord. Root ganglia of spinal cord. Okay, spinal cord. Clear? Then one more we have, students, multipolar neuron. That is the neuron which I drew here. Okay, this is a okay, where is it? This is a multipolar neuron. That means many dendrites are there, many axon endings are there. This is a multipolar. Got it? I'm writing it here only. This is a multipolar and this is the most common. Okay, most common, found almost everywhere. Found almost everywhere. Got it? Chalo. Yes. Clear? Any doubts? Types of neuron? Okay, types of neuron. We have unipolar neuron, bipolar neuron and pseudo unipolar neuron. One is single, you can see single axon. Okay, one axon. One axon. Here what is happening? One dendrite, one axon. Here also one axon, one dendrite. But this is initially forming, it is in one extension. Yes, brain, multipolar neuron is present most of the places. Okay, okay, most of the places. Any doubt? Any doubt? This is clear? Okay, look here students, here also you can see bipolar neuron, you can see one dendrite, one axon. Here also you can see pseudo unipolar, okay, multipolar. See, actually this diagram is not exactly right, but if you look here properly, it is one emerges like this. See, can you see? One is emerging, one is emerging. But look here, from here like this it is emerging. Okay, don't get confused seeing that ma'am you told it's not multi, how come this is multi? No, you can see that from one branch itself it's just splitting. Okay, difference is there. Same way here, you do not have pyramidal cells, it is everywhere scattering is there. Okay, everywhere scattering is there. Ready? Done? Easy peasy lemon squeezy. Any doubts? Okay. What is happening? Yes, okay, done. Now, electrical and electrochemical transmission. That means neuron to neuron, what is happening? If neuron to neuron there is a space, what is going to happen? That is what we are going to understand. Okay. Now students, very simple. Electrical and electrochemical transmission means, students, 1 second, let me see if I have, 1 second, I'll make a copy of this. Okay. Look here now all of you. This is a neuron. Okay. This is a neuron. I wish I could copy this, but I can't. This is another neuron. Okay, this is another neuron. This is another neuron. Can you see 3 neurons are arranged? Okay, might not look like a neuron, but it is a neuron. Okay. Trust, that is everything. Should trust your teacher. Now look here. One signal has been sent. Okay, one impulse has come. One impulse has come. Now this impulse will be transmitted from the dendrite to the cell body to the to the axon, and it will continue. Okay, it will continue. Now students, look here, from one neuron, that is from the dendrite head. Okay, always remember your signal will be sent from the head of one. Okay, this is the head to the tail of one. Okay, head to tail. This is head where your dendrite is the tail of another one. So don't think your neurons will be like this, your neurons will be like this. Okay, they will be, let's say, like this. Okay, another one's tail will be like this, head will be like this. It will be circle type. Okay, it will be circle in a loop region, it will be there. So this is not how your neurons will look like. But you understood? Let's say this is the head. Okay, this is the head. Tail of this neuron will come here. Tail of this neuron will come here. Got it? Okay, tail of this neuron will come here. Got it? So now students, between these neurons, what is going to happen? An electrical impulse is going to be passed on. Correct? From here one impulse came, it will be passed on, passed on, passed on. Here you can see there is a gap. In the gap, what exactly happens? Wait 1 second. This is going to be an electrical impulse. Okay, this is going to be an electrical impulse. Okay, ma'am, understood. One impulse came, ma'am. It went all the way through my axon, through my dendrite axon. Okay, understood. Now, between these two axons, there is a small gap that you see. Okay, there is a small gap that you see. If a gap is coming, then what will be happening? If a gap is coming, it is going to be a chemical, okay, chemical impulse or a chemical transmission. Okay, let's write chemical transmission. That means, students, whose use are we going to take? Okay, we are going to use the neurotransmitter. We are going to take the help of a neurotransmitter. Is this clear? we're going to take a help of a neurotransmitter. Same way here also, what is going to happen? An electrical impulse will happen. In an axon, an electrical impulse will happen. Okay? Electrical impulse. Again, chemical transmission will happen. Okay? Chemical transmission will happen. Again, electrical impulse will happen. This is how your signal is going to get transmitted. So, wherever electrical impulse is happening, this is going to be at a very faster rate. Very faster rate. But wherever the chemical transmission is happening, there can be a slight delay. Because why? This neurotransmitter has to be released, it has to be accepted by the receptor and it has to go. Yes, very good. The gap should be, okay, for a chemical transmission to happen, a gap should be more than 2 nanometer. Okay? Gap should be more than 2 nanometer. Got it? Got it, all of you? Gap should be more than, yes, chemical is comparatively slower than your electrical. Okay, obviously students, something has to open, okay, something has to open, then only another thing can be received. Okay? Now, all of you understand this very simple thing, that one impulse is coming, okay? Impulse has come. This impulse will be passed on to the dendrite. Okay? From the dendrite, it is going to go where? To the cell body. Okay? To the cell body. From the cell body, it is going to go to the axon. From axon to axon ending. Okay? Axon terminal. Okay, this is how the passing is going to happen. Impulse came, dendrite took it, dendrite passed that information to the cell body to axon, axon to axon terminal. From an axon terminal, it will go to another dendrite of another neuron. Okay, dendrite of another neuron. Understood? It's always like this. Okay, it's always like this. This is the head. Okay, this is the tail. This is the head of another. This is the head of another neuron. This is the tail of that neuron. Got it? Tail, head, what I'm talking about? I'm talking about a dendrite. Okay? So, this becomes the dendrite of this neuron. Okay? This becomes the tail. Okay? This becomes the tail. That is your axon. Again a dendrite and again an axon. Okay? Another axon. Got it? Head to tail. Always remember this. Many students do not understand because all of us draw the neuron straight. But that's not how it is. Head to tail, head to tail, head to tail. That's how it continues. Okay? Chalo. Now students, I was supposed to write it that side, but it's okay. I'm writing here the neuron. What are the two portions of the neuron? Okay? Let's first separate it. Here I'm writing. See, when we learn the neuron, okay, two parts are there, no? Okay, what is going to be there? Cyton or your cell body or soma or perikaryon, many names are there, okay? Perikaryon, got it? These are the names of your, or cell body, okay? Cell body. Here you have two portions, okay? Axon, so wait. Long unbranched, okay, long unbranched portion called the axon and the short branched portion called the dendrite. Okay, branched, students, full I cannot write, maybe I have to write it more small. Give me 1 minute. Okay, I'm writing it here. Cyton, soma, perikaryon. These are all the names of the cell body. Okay, different names are there. Cell body. Okay, then here you have the next part of the neuron, that is your branches. You have a long UB, I'm writing unbranched. Okay, long unbranched portion called the axon. Is axon branched or unbranched? Axon is branched or unbranched? Unbranched. No one has a doubt in that. Now, this is, we have a short branched portion, okay, called the dendrite. called the dendrite. Clear? This I was supposed to write it that side, but it's okay. I've written it here. Okay. This is long. Okay, dendrite. Students, who receives the information? Who receives the information? Yes, very good, very good. Who receives the information? I think chats are slow here. There, yeah, unbranched. Good, good. Perikaryon, yes, very good. Who receives the information? Tell me quickly in the chats. Fast, fast. Who receives the information? Yes. Fast, fast. Very good, very good. Now the chats are seeing dendrites, right? Dendrites. Very good. Awesome. Okay. Awesome. Now, students, we are going to the major portion. Okay, major portion of this chapter, action potential. Right. Now, students, we have understood neurons. Okay, we have understood what are the types of neuron, different types of neuron. We also understood what else did we do? See, we already finished so many slides. We understood what is the difference between your coordination, that is your endocrine, and your nervous system. We understood the different types. We understood the neuron diagram, different types of neuron, again different types of neuron, electrical impulse and chemical transmission. Now we are going to go to the action potential, most important. Okay, most important portion ever. Got it? Now students, look here. Now the neuron gets an impulse, right? It has to generate. Okay, it has to generate the impulse. Now for generation of the impulse, there should be a difference. So that is why where you come across the word called as generation of impulse. Okay, generation of impulse. That is, if an stimulus has come, how do we react to the stimulus? Now a dog is coming to bite me, right? My nervous system got the information and immediately I will start running. So here what happens? Your nervous system is working, your muscles are working, locomotion and movement, all of you studied, right? Now we are going to see how the generation of an impulse is created. This electrochemical, instead of this you write generation of the impulse. Okay, let's draw one, how the axolemma and the axoplasma looks like. Okay, axolemma and the axoplasma looks like. Look here, here. Okay, done. Okay students, this is two membranes. Okay, two membranes you're going to see. Okay, now this is going to be what? This is going to be what? Can anyone tell me in the chat? Is it the axolemma or is it the axoplasma? Who is axoplasma? Who is axolemma? They'll be like, ma'am, why are you asking us? Okay, students, this one is the plasma membrane. So this is going to be the axolemma. Yes or no? This is going to be the axolemma. Axolemma. Okay, this is going to be the axoplasma. Cytoplasm, no? Axoplasma. Okay, clear? Now, carefully listen to me. Carefully listen to me. That's when I realized I have to drink water. These are your two membranes. Okay, axolemma, axoplasma. Yes, very good. Outer, you should tell like that. Always you should tell outer and inner. Now students, this outside one, there is a fluid. Okay, that fluid will be called as extracellular fluid. What is it called? ECF. Okay, extracellular fluid. Extracellular fluid, ECF. So if this is ECF, can I call this as ICF, that is intracellular fluid? Agree or disagree? If this is extracellular fluid, then the one inside is going to be ICF. that is intracellular fluid. Now, in ECF, that is extracellular fluid, there is a rich sodium content. Rich sodium content. Okay, that is here it is going to be positive. Okay, outside it's going to be positive. And this positivity is because of whom? Because of the sodium ions. Because of the sodium ions. Students, what am I drawing? I am drawing a polarized stage or a resting potential. We can call it as RMP. Okay, or I will call it as a polarized. Okay, polarized state. And this polarized state voltage is going to be around minus 70 MV. Okay, minus 70 MV. Okay, polarized stage means what? Both the side ions are there, and both the side, like imagine I am also happy, you are also happy. We will never have a fight, right? Why will we fight? Imagine I am angry and you're calm. I'll definitely get angry that why he is not angry or why she is not angry. I am angry, why are they not angry? That means there is a difference in the ions. Got it? Same way, imagine both the sides. Positive is there, negative is there, both are polarized. Polarized means both the side ions are there. Okay, both the side ions are there. Got it? Chalo. Here students, positive ions are there, that is sodium ions are there. Inside what do you expect? Tell me in the chat, all of you. Inside what do you expect? Obviously if that side is positive, this side should be negative, ma'am. It's understood, right? This side should be negative. Okay, let it be that. Now I'm going to draw some channels on them. Okay, I'm going to draw some channels on them. Students, these are open channels. Okay, the membrane of our axon will have open channels. Okay, open channels like this. Okay, these are your sodium channels. Okay, these are your sodium channels. The same way they are going to also have potassium channels. Okay, they are also going to have potassium channels. You can draw it like this or you can draw it in another way as well. Okay, there are potassium channels as well. 1 second. I'll draw potassium channels like this. Too much task, students. Let's draw this slowly. All are same size, but when we draw, it becomes different size. These are potassium channels. So on the axolemma, okay, on the axolemma, there are some sodium channels and there are some potassium channels. Okay, there are some potassium channels. Okay, these are your sodium channels. Now students, it is very simple. Okay, we understand one thing. Whatever, be it a channel and it is always open, it is working passively. Now since it is working passively, you people tell me, will they allow the movement? From where to where will the movement of sodium happen? From outside to inside or inside to outside? Okay, outside to inside or inside to outside? Tell me quickly. Down, negative is there. I'll tell you about the negative. Outside to inside or inside to outside? Tell me fast. More times if I ask, I'll only get confused. It will allow influx of sodium from outside to inside or inside to outside. Quickly in the chat. Fast, fast. Out to inside. Okay, very good. Very good. These are open channels. Okay, two channels are present. Let's go once again. Two channels are present on your axolemma. Okay, and they're always open and they're leaky channels. Very importantly, these channels are leaky. So that means something or the other keeps coming in and going out. Both the channels, sodium channel as well as the potassium channel, is leaky. Okay, and they do passive exchange. Passive exchange means something will move from a region of higher concentration to a region of lower concentration. That means sodium, which is rich in the outside, right, in the ECF, will definitely start moving inside. Okay, will definitely start moving inside with the help of this channels. So here automatically sodium will start coming in. Now what about potassium channels? All of you tell me in the chat. What about the potassium channel? Outside to inside or inside to outside? What about the potassium channels? Quick, okay? Fast answer. Potassium channels, outside to inside or inside to outside? Where it is more? Okay, yeah, it is chemistry class. I have to teach all the subjects, okay? I have to teach all the subjects. That's where we have reached. Jaldi. Very good. Ma'am, from inside to outside, because obviously here the concentration is more. So, it will go outside. Shall I draw it with another color? Okay. Inside to outside, inside to outside, inside to outside. Now students, if you look at this diagram, you will notice two things. You will notice that sodium channels are comparatively lesser, right, and potassium channels are more. So that means more potassium will go outside, okay, more potassium ions will go outside, will move to the ECF. More potassium ions will move to the ECF. Can you see? I did not draw by mistake. I drew actually more. Okay? I actually drew more. Right? That means they have more potassium channels and more potassium will go outside. But here what is happening? Less sodium intake will happen. Less sodium influx will happen. Okay? Less sodium influx will happen to the ICF. Is this clear to all of you? Is this clear to all of you? Now, in such a situation, okay, in such a situation, we need someone to save, right? We cannot allow this to happen. Okay, we cannot allow this to happen. Why? Because if I want to achieve resting potential, my axolemma and this should be having positive outside and negative inside. But here I notice that this potassium, when it's moving outside, more is moving outside, and sodium, when it's coming in, less is coming in. This will not allow it to get polarized. So for that we have sodium potassium pump. Okay, sodium potassium pump. This sodium potassium pump, students, will be present here somewhere. Okay, it will be present here. Okay, it will be present here. Okay, they will be present here. The sodium potassium pump, I am extending this, okay. Excuse me. So sorry. So here there is a sodium potassium pump. I think the colors are too much, okay. This is a sodium potassium pump. So in this one will work on energy. Okay, that is, this sodium potassium pump will utilize 1 ATP. Okay, it will utilize 1 ATP, and what is going to do? Okay, what it's going to do? It's going to take in 2 potassium ions and give out 3 sodium ions. Okay, it will give out 3 sodium ions and put in 2 potassium ions. This way what will happen? We will be able to reach the polarized state. Agreed or disagreed? Okay, we will be able to reach the polarized state. That means it will take 1 ATP and it will give 3 sodium outside and 2 potassium inside. Okay, 2 potassium inside. This way we will be able to reach the polarized state. Otherwise we will not be able to reach. Okay, otherwise we will not be able to reach. Is this clear to all of you? Yes or no? Tell me in the chat box quickly. And students, understand? Yes, very good. Understand, permeability of potassium is more. Okay, this is the line of your NCERT. Permeability of your potassium ions is comparatively more than sodium. Is more than sodium. Okay, more than sodium. Yeah, such conjuice. It's okay. Is this clear? Any doubts? Now, students, one very important thing that I have to tell you people is that this Potassium, I've written positive sign. Ma'am, but you're continuously telling that inside it is negative. Understand, the negativity is caused because of some proteins. Because of some proteins. Negatively charged proteins. This negatively charged proteins allows the positivity. Okay, that positive charge, no, is because of this negatively charged protein. Sorry, not positive, the negative. Okay, sorry, the negative is because of the negatively charged proteins. Potassium is not enough to show the charge. Is this clear? So this negatively charged proteins will not be able to do this exchange. Okay, potassium sodium ions only will be able to do, but this negatively charged protein is the reason for this negativity in the axoplasma or in the ICF, okay, intracellular fluid. This you have to know. Proteins, negatively charged proteins. Clear? Yes, exactly. Okay, exactly. So that's the thing. Now students, I have another amazing diagram to explain you. Look here, all of you. This is how action potential will be created. Now what did I show you? I showed you the resting potential. That means where the sodium will be there, that side will be positive, and where the potassium will be there, that side will be negative. This is the resting potential where the voltage will be minus 70 MV. Now one impulse has been created. One impulse has come. Let it be like you touched a hot object. Let it be like you went somewhere, whatever. One impulse has come. That impulse should have a threshold capacity. That means if the impulse is not enough, having enough threshold, it cannot make a change. It cannot move this resting potential. Now imagine I hit you. Okay, I hit you and I hit you so softly that you didn't even feel it. You're like, that means my hitting, right, that the amount, the energy with which I hit you was not the threshold. Okay, was not the threshold. So that threshold capacity has to be reached, or the threshold value has to be reached for an impulse to be created or an action potential to be created. Got it? Now, when an action potential is created, what is going to happen, students? Immediately the sodium pump will open. Okay, sodium pump will open and sodium will start moving towards the inside, to the ICF. Okay, now can you see what happened? Here it became negative charge. Obviously, sodium is less means there will be negative charge. Now, all the positive went down. So, here it will become positive. This will be called as depolarization. This will be called as depolarization. Okay? Yes, this is the physics class. Okay? Now, once the sodium reached here, the sodium channels closed. Sodium channels closed. Let's say the threshold has to be at least minus 55 MV. Okay, minus 55 MV, it can go up to plus 35 MV or 45 MV. Okay, in some one book, one is given. Okay, now students, the action potential was created, the sodium channels opened and the sodium went to the potassium, to the ICF, and here it caused a positive energy change happened. Okay, chalo. Now, what has happened? Now, after some time when the sodium channels closed, the potassium channels opened. This will let the potassium to move that side. Okay, now obviously here it is going to become positive, here it is going to become negative. Okay, here it is going to become negative because here only proteins will be left, right? Only some negatively charged proteins will be left, they will be overpowering the positive. So here it will be negative. This will cause repolarization. Repolarization, that means you are going to become again back to polarized. Okay, we are going to go back to polarized. Got it? Yeah. Okay, now students, what is going to happen? After one point, our sodium potassium pump was there, no, to solve all the issue. They will open and they will start taking 3 sodium and throwing it this side, taking 2 potassium and throwing it this side, and we will go back to the resting potential. Now, I have a very funny way to remember this. Okay, I think some of you who have attended my last year's class already knows it, but for those who are new to this class, I'll give you a very funny example. Okay, funny example. This is just to remember this diagram. Don't use this in the exam. Like, don't write it in the exam. Okay, this funny example is this. Okay, this is you and this is your sibling. Sodium is you, so potassium is your sibling. You both are happy, but suddenly your mom or dad bought a piece of cake or gobi manchuri or chicken or something they bought that you people love the most. That is the impulse. Okay, that is the impulse. Now, when the impulse got created, what happened? You guys got angry because the division was not equal. Okay, your sister got more or you got less, whatever. Now, your sister got very angry and your sister will move. Okay, your sister will move towards you to attack you. Since your sister is attacking you, she is going to become the negative person. Obviously she's going to become the negative person. Okay, you did not attack, you're very calm, because maybe you got the bigger piece, you're very calm. Okay, that means when your sister is attacking you, you're in a depolarized state. Depolarized state. Okay, she'll punch you, you'll become depolarized. So she'll come, the gates will open and she will move towards to attack you. After some time she'll find peace. She'll be like, hitting this buffalo, no use, it's this girl is not moving or this boy is not moving, I will just stop. Okay, and the sodium channels close. Okay, now what will happen? You will lose your temper. Okay, till now you were passive, you're like, it's okay, it's okay. Now you will lose your temper. Now when you lose the temper, you become the negative person and you will move to attack her. You'll go to her territory to attack her. Since she is not attacking, she'll become the positive, you will become the negative. This will become a repolarized stage. Okay, repolarized stage. Now, your voltage or your sodium potassium pump is there, no? That is your parents. They will come, they will take your sister and throw that side, they'll take you and throw this side. This way what will happen, we'll go back to the polarized state again. This is the funny example, okay? Please let me know how the example was, okay? Yeah, Raksha Bandhan special example of your sibling and you. Has the students, all this happens in all of our houses, yes or no? First they will attack, or first we will attack, then they will not attack, then after some time they will attack. By then everyone, the whole neighbors will get to know that, my God, what these two kids are doing. Then they will come, then they will separate us and they will go back to the polarized state. So during depolarization, you're getting the hittings. Okay, during depolarization, someone is attacking you. Who is that someone? The sodium pump has opened and they are coming. Okay, since you're not attacking, you will be in the positive. Since they are attacking, they'll be in the negative. Then again ulta will happen. Now you will go there and attack them. Right, that means potassium pumps will go. Since you, imagine if you are the younger one, you have more permeability. Okay, that means your parents will have more attachment to you, or more, they'll be like, Pavam, she's small, no, leave it. Pavam, she's small, no. That permeability is more for the potassium. Okay, so potassium will go and attack. Then neighbors will come, other auntie, uncle, everyone will come and separate you. That is your voltage pump, sodium potassium pump. Is this clear? Yeah, moral of the story is happy Raksha Bandhan. Yeah. Okay, is this clear or not? Tell me, this image cannot be done in a better way. Okay, image cannot be done in a better way. Now look here. See? Negative, positive, negative, positive, negative, positive, negative, positive. Can you see? This is how an action potential will be created. From here an impulse has come. From the cell body, see how it is moving through the axon lemma. This is what I drew. Okay, axon lemma. Now it is going to the axon. Okay, wait 1 second. Now students, here can you see? This saltation I'll teach you. Now you can see how it's jumping from one myelin sheet to another wherever the axon is exposed. Now look here. Action potential. Okay. Then here what is going to happen? Repolarization, depolarization. So here also you can see the signs changing. This I'm going to teach you now. This I'm going to teach you now. Is this clear to all of you? Yes or no? Clear? Any doubts? Yes, students, elder one's life is little hard. Okay. Chalo. Ready? Okay. Okay. Now what I told you. See, I made you understand what is going to happen. So now you understood. Now you tell me what is going to happen. Action potential will be created. That means an impulse is coming. Okay, an impulse is coming. When an impulse is coming, what is going to happen? First, your students, this is going to take some time to draw. Okay, I'll draw it in just one. Okay, I'll just draw one line. You should understand that both the layers ma'am has drawn. Okay, I'll just draw it in one line. I think I won't draw it down. Okay. Done. Okay. Look here, all of you. First, when an impulse is coming. Okay. First, when an impulse is coming, let's say, okay, no impulse is there. Let's say this is the normal one. Here, positive is there. Okay. Positive, 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 positive. And this is because of your sodium ions. Okay. Down we have negative, 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 negative because of your potassium. Is this clear? This is a RMP state or a polarized state. Okay, polarized state. Students, here what will be the voltage? Here it's going to be minus 70 MV. I hope no one has doubt in this. Minus 70 MV. Yes? Tell quickly. Any doubts here? Okay. Now, a stimulus has been created. Okay, let's erase the stimulus. Let's make another stimulus here. Okay, here students, one stimulus has been created. The stimulus has to, okay, impulse. This impulse should have threshold value. Without threshold value and from the resting potential, we cannot change it. So, it can be from minus 55 MV to 65. Okay, 65 MV. Some places you will see 45 MV. Okay, this is one question mark. Okay, 45 MV. Now, students, one impulse has been created. Immediately, who is going to open? Your potassium channels are going to open and they are going to influx. Okay, so let's say potassium was here. Now, influx will happen. Okay, influx will happen. Okay, now what is going to happen? This side, right? This side became positive. This side became positive. Here, everywhere it was negative. Where the action potential got created, there you can see a sign difference. Can you see here? Can you see here? Here it became positive. So this will lead to what, students? This will lead to re or depolarization. Re or depolarization. Quickly in the chats. Jaldi, jaldi. D, re, D. Tell fast. Depolarization. Yes or no? These people have to tell. Tell fast, students. I'll wait for your answers. Then later you'll tell me, ma'am, you did not wait. Yes, very good. Depolarize. Some is telling re, some is telling D. Depolarization. Okay, depolarization will happen. So during depolarization, what happens, students? The first thing that will happen is your sodium gated channels will open. Okay, channels will open. Sodium will happen. Influx of sodium will happen. That will cause a, with sign, that will cause a positive sign on the down portion, right? Let's say this is your ECF, this is your ICF. In the ICF, you will start seeing a positive sign, and on the ECF, it will be a negative sign. Okay? Yes. Chalo. So here, positive sign will be seen. Okay. Now, students, influx has caused. Okay. Now, what is going to happen? Reversing will happen. Right? Reversing will happen. This will lead to what? Here, now, the potassium channels will open. The potassium channels will open and they will start moving out. And when they move out, always remember, they will move out more. Okay. They have more channels, so more moving out will happen. Okay, more moving out will happen. So here the potassium influx will start coming in. Okay, potassium influx will start happening. Okay, this will lead to what? Here it will become negative. Okay, this side will become negative. Okay, if I write negative here, it will become confusing. Okay, here potassium, I'll just write this one, okay, I'll just write here potassium gated channels open. Okay, channels open. Students, here it is going to be negative, I'm not writing it here. Okay, here it is going to be negative because why? Potassium was positive, that also left that side. Now only the negatively charged protein is going to be there. Here it is going to be positive, okay. That side is going to be, here is going to be negative, that side is going to be positive. So here potassium gated channel open and obviously influx of, okay, efflux of, not influx, efflux. Efflux of potassium happen. Just think about the fight I told you. Think about your sibling example, that is more than enough. Okay, more than enough. Okay, efflux of sodium will happen, I mean potassium will happen, and because they have more channels, more more efflux will happen, okay? More efflux will happen. Now, how do I fix this? How do I fix this? Siraj, thank you. How do I fix this, students? Tell me. This will cause what? This will cause what? Ma'am, repolarization will happen, ma'am. Okay, this has caused repolarization. That means we are going to become polarized. So, repolarization will happen. After this situation, what is going to happen? The gated channels are going to open. That is, your sodium potassium pump is going to open, and this will, the pump will do what? Students, I am writing that also here only because there is no place. Okay, I am writing the pump here. This pump will do what? The pump will simply use one ATP and throw 3 sodium outside, okay? 3 sodium outside and push 2 potassium inside. Push 2 potassium inside with the use of 1 ATP. Is this clear? Clear? Any doubts? Any doubts? This way we'll be able to go back to polarized stage. Okay, we'll be able to go back to polarized stage. Is this clear? Is this clear? Yes, very good. 3 Na out, 2 Na inside. Got it? Okay. Those who did not see the first example, it's going to be difficult for you to understand. Otherwise, it is easy. Okay. Otherwise, it is easy. Okay. This is generation and conduction of impulse. Yes, exactly. Again, resting potential after repolarization. Okay. It's very simple, students. You shouldn't complicate in your brains. If you complicate it, then it's gone. Okay, if you complicate it, then it's going to be difficult. Okay, if you complicate it, it will be difficult. Now the question comes, ma'am, okay, I understood that this is happening, but how is it transported through the entire axon, right? Because now I just showed you exchange, up and down exchange is happening. How is it transported through the entire axon length? That is a question. Yes or no? That is a question to all of you. That question I am going to answer with the help of one diagram. Okay, all of you stay tuned. Stay tuned. Sorry. Okay, look here. One portion. This is one portion. Okay, same way. This is one portion. This is one portion. Okay, let's first draw 2 and learn it. Okay, now students, look here. I am going to split this. You have to think and tell me, okay, which portion what might be happening. Okay, look here, students, we are going to split the neuron into 3 parts. Okay, how it is going fully, okay, how it is getting transported fully is the question. So look here, this is going to be A, okay, this is going to be B, and this is going to be C. Same thing here, A, B and C. Now these are the 3 sets of your neuron. Okay, don't get confused, just listen to me carefully, don't take your attention. Okay, normally what happens in a polarized state, right? In a polarized state, outside is going to be positive, right? Outside is going to be positive and inside is going to be negative. Anyone has doubt in this? Outside is positive, inside is negative. Anyone has doubt? Take screenshot of GM Express. There's no GM Express for this. GM Express only with mouth, right? GM Express, I'm writing everything again. What will I write? Okay, yes. So students, look here, all of you. This is the first, this is your RMP, right? We have reached the polarized state here. This is the polarized state. Okay, neuron has reached the polarized state. Okay, ma'am, polarized state. This portion also is going to be polarized. Okay, plus plus, 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 minus, 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 again plus. This is a normal resting neuron, okay? Excuse me. Okay. Oh, GM Express of last class. Okay. Students, this is the polarized state. All three are in polarized state. Okay, I'm writing here RMP. Okay. This is also RMP. Now, one threshold, that is one impulse has come. Okay, one impulse has come here. Threshold impulse. Okay. If it is not threshold, then it cannot change the resting potential. Threshold impulse has come. What is going to happen in A? Can anyone think and tell me? Okay. Can anyone tell what is going to happen in A? Think and tell. Obviously, if an impulse is coming, definitely change of signs is going to happen. That is, outside is going to be positive and inside is going to be, sorry, outside is going to be negative and inside is going to be positive. Look at this, compare this. If you understand this, then there is nothing else, okay? Everything is easy peasy. Yes, students? Here what has happened? Here depolarization has happened. Yes or no? Depolarization has happened. Now, here what is going to continue? This portion what is going to continue? Obviously, here also RMP only is going to continue because here nothing has happened. So, here it is going to be positive, 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 negative, 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 negative. Here also same thing, positive, 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 negative, negative, negative. What I am trying to show you, I am trying to show you how an impulse is transported through the entire length of an axon. From here it starts, then it moves, then it moves. That is what we are going to understand. Okay. Now, students, this is a normal axon. That is, this is in the resting. Okay, resting stage it is. Now, from the resting stage, here an action potential was created, right? Here an action potential was created. Okay, here action here an action potential was created when a impulse came. Okay. Now, this became depolarized. Can you see? Signs have changed. Now, what do you think next will happen? What do you think next will happen, students? I have to move this impulse, right? I have to move this impulse from one place to another. So, what is going to happen here? Very simple. Remember that in the outside, okay, outside of the this is axolemma, the ions are going to shift like this. They are going to shift like this. That is, they are going to move from B to A. Inside, they are going to shift like this. They are going to shift like this. Can you relate this? Okay. Outside, very simple. Outside, it's going to move from what to what? B to A. B to A. Okay. Inside, it is going to shift from A to B. A to B. This is a very, very important topic. Okay. Those who did not understand, ask me an explanation again, but do not skip this topic. This is how the transport will happen till the end of the axon. Okay. Till the end of the axon, this is how it is going to happen. That is, this will move here, this will move here, same way this will move here, this will move here. Like this, the charge will keep continuing. Charge will keep continuing. Okay, clear? I'm sorry for this one, but what to do, students? Cold. Is this clear? Yes or no? Okay. Now what is going to happen in the next situation? This is going to become positive. Down it's going to become negative. It will become repolarized, right? Repolarized. There's no place to draw it again, but I hope you understood this. Okay. I hope you understood this. Okay. Yes. So this will shift here. So obviously this is going to become positive. This will shift here. So obviously this is going to become positive. Like this here, what is going to happen? This will go to, so this will continue, and here what is going to happen? It will become positive, 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 negative, negative, negative. But here what is going to happen in the next stage? This shifted there, this shifted here. So this will become negative, 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 positive, positive, positive. So this will go through a repolarization, this will go through a depolarization, this will keep continuing. Clear, right? Clear, right? Nothing is there in our hands, sir. What happened, Sony? Depression, huh? You want me to draw one more, huh? Down? One more might not fit. I should have drawn it a little more small. So it will go from re to d, d to again this. So this will become, wait, let's try to draw one more. I think then you will understand it more better. Students, I'll try to draw one more here. So that time you can tell me. Okay, ma'am, yes, we understood this. Okay, let's draw one more here, one more axon here. Okay, again one more here. Okay, it's kind of straight. Okay, this is the third one to show you. Okay, look here, students. Now think of this and tell me this. Okay, think of this and tell me this. What is going to happen here? What is going to happen here? This depolarization will go to repolarization. Yes or no? So here this has shifted. So this is going to become positive, 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 negative, negative, negative. This will go to a depolarization. This is going to a repolarization. Okay, repolarization. Now from repolarization, what is going to happen? Here, your pumps are also there. Okay, sodium, potassium pump was also there. That you shouldn't forget. Okay, from here sodium will start coming in. This will lead to what? This will lead to what? Obviously, this will go back to this situation, that is negative, 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 positive, 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 positive. Okay, this will become a depolarized state. This portion of the neuron will still be in RMP only. Okay, no changes here, no changes here. They will still continue to be in RMP, that is polarized state. Is this clear? Any doubts here? Just look in, tell me. Very simple. From outside to inside. See, this positive went here. So here it became positive. This positive will go here. Again positive. Okay. Same thing. This positive came here. So can you see positive here? Okay. And this way same thing will happen. Again positive here. So from repolarization this became depolarization. But this portion, the C part of the neuron still remains in RMP. Okay, C part of the neuron still remains in RMP. So this is how the movement will happen. So what you have to do, you have to know this. You have to know this. Clear? I want thumbs up in the chat box. If you understood, thumbs up in the chat box. Yes, students? Yes, quickly. Yes, got it, all of you? Okay, done. Okay, this is how it will happen. Now I'll show you that GIF you will be able to understand. Okay, students, look here. See, now impulse is coming. Look here. Positive was there, negative, negative, positive, negative, positive, negative, positive, negative, positive, negative, positive, negative, positive. Understood how it is coming? So it's going to depolarization, repolarization. Okay, that is what is happening. Clear? Negative, positive, negative, positive, negative, positive. This is how it will move. So this portion is the A portion of the neuron, this is the B portion, this is the C portion, this is the D portion. When A, it is moving from outside, how it will go? B to A it will go, B to A it will go, inside A to B it will go. Okay, from here to here it will go, this way, like this. Okay, because here this side I have to make it back to polarized, no? So obviously I'll move the signs like this. Okay, same way. Where's the remote? Yeah, same way this one also. See here, can you see how they're jumping? See, they'll move like this. Can you see how it's jumping from B to A? Okay, same way A to B. See here also you can see how they're moving. I hope it is clear. Okay, this is how it will happen. If you understand this, then there is nothing much difficult, students. Okay, we'll just see NCERT as well. There is one graph. Okay, there's one graph to understand. This will just help you understand the threshold. So 70 is the resting potential, bro. This is the RMP. Okay, this is the RMP we were talking. Here no impulse, nothing is there. Now impulse, what has happened? Here impulse has been created, that is the threshold value is minus 55. The moment threshold comes, obviously depolarization will start and then they will reach an action potential. Okay, then again repolarization will happen. Okay, repolarization will happen. Now at some places what will happen, students, like this will happen. This is a hyperpolarization. Hyperpolarization, that is around 90, minus 90 MV. Okay, hyperpolarization will happen. That is too much influx has happened, that will lead to hyperpolarization. Got it? Minus 90 MV will cause hyperpolarization. My God, in my life this much physics I haven't learned. Okay, so is this clear? Is this clear? Very simple. The threshold impulse has come, depolarization to re. So it will go like this. It will go like this. RMP, that is polarized state, to first D will go. Okay, depolarization, from D to re. Sometime from re to hyperpolarization it can go. Otherwise it can go again back. Hyperpolarization, again we'll continue the same cycle. Okay, same cycle. It will again come back to RMP. From RMP, again the same thing. Okay, so here depolarization is happening because of a stimulus. Okay, stimulus. Then it is getting carried away. Again from here one more stimulus comes, impulse comes, then this will again continue. This is the cycle. Clear? Yes. Hyper resting phase again. Very good, very good. Okay, hyperpolarization, then resting. This is again not necessary every time it happens. Okay, not necessary every time it happens. Students, clear? Okay, clear? Here is where your gated channels will open. Okay, gated channels will open so that they can throw these people and put them back into control. Okay, now we'll go to this one and then we will stop the class for today. Synapses I'll take next class. Okay, synapses, brain I'll take next class. Fine? Okay, chalo. Now look here, you might be curious to know about the mechanism of generation of nerve impulse and its conduction along an axon. When a stimulus is applied, okay, when a stimulus is applied at a site, okay, applied at a site on the polarized membrane. Polarized membrane means minus 70 MV. Okay, polarized membrane. The membrane at the site A becomes freely permeable. Very important. The site A becomes freely permeable for sodium. Till that, understand, when the resting potential was there, the membrane is more permeable to potassium, less permeable to sodium. Younger sibling always gets the benefit. Okay, so it is more polarized to the potassium, not to the sodium. But the moment an action potential is created, the membrane will become freely permeable for sodium. This will lead to rapid influx. See, very important. This will lead to rapid influx of sodium followed by reversal of the polarity at that site. Look at the diagram that I drew and try to understand. I will read, you listen. You look at the diagram. Okay, rapid influx of sodium that allowed the reversibility. That means what was on the positive was there, down negative was there. They got reversed. Top negative came, down positive came. Okay. And the outer surface of the membrane becomes negatively charged and the inner side becomes positively charged. Okay, some dark color I have to take, students. Okay, so the outer surface will become negative, okay, and the inner side will become positive. Okay, now the polarity of the membrane at the site A is thus reversed, hence it is called as depolarized. Okay, hence it is called as depolarized. Done? Okay, ma'am, understood. Now the electrical potential difference across the plasma membrane at the site A is called as an action potential. So when an impulse is coming, that will create a difference, that will cause the action potential, okay, which is in fact termed as a nerve impulse. Simple. Okay, now at this site, at sites immediately ahead, the axon, that is B side, which I drew, no, membrane has a positive charge on the outer surface. Normal thing, positive charge on the outer surface and negative charge on the inner surface. Are you all looking at the diagram? I will read, just look at the diagram. Okay, result a current flow on the inner surface from A to B. See, that is A to B it will go. Okay, inside, inner surface A to B. Now, on the outer surface, the current flows from B to A. Students, from NCERT itself, I am teaching you, okay? I am not teaching you from anywhere else, from NCERT itself. See, you can see all this just now I taught you. Outer surface, the current flows from side B to A. To complete the circuit of current flow, hence the polarity at the site is reversed and an action potential is generated at the site B. This is how it goes all around the axle. How does a gossip go? Now when gossip is told, it will go from one person to another person, another person to another person, another person to another person. Yes, hi Angeline, I'm doing good. So it will keep passing on. Same way, A to B it will go, B to C it will go. Same way, what will happen? Repolarization, depolarization, repolarization, hyperpolarization, again resting potential, again continues. Okay. Thus, an impulse is generated at site A, arrives at site B. Arrival has happened. Okay. Now, the sequence is repeated along the length of the axon and consequently the impulse is conducted. The rise in the stimulus induced permeability to sodium. See, this is important. Starting it was not liking the sodium, okay, later it started liking. Stimulus induced permeability to sodium is extremely short-lived. Short-lived, because if it lives for a longer time, then more action potential will be created, that we don't want. So it is quickly followed by the rise in the permeability of potassium. That is, students, the moment they tell, oh, my elder daughter is good, my elder son is good, they will get 2 horns. So immediately it will drop down. That's what they're trying to say. Within a fraction of seconds, the potassium diffuses outside the membrane and restores the resting potential of the membrane. So potassium will be able to do that because it has more channels. It has more channels and it can move faster than sodium. Right? More channels are there, means more way for it to go out. More way for it to go out. So potassium will do that. That is the reason why permeability is there. Okay, permeability is there. Okay, yes. Now potassium diffuses outside the membrane and restores the resting potential of the membrane at the site of excitation, and the fiber becomes one more responsive to the further stimulation. Okay, stimulation. This is the exact thing. Is this clear? Myelinated. Okay. So in this, if you understand, nothing like that. Okay, this you should understand, and this you should understand. B to A to B. Then, can I actually? No, I cannot. Okay, this one and then this one. Okay, this one, if you understand, you are clear. Okay, here what is happening? Here both the channels are closed. Okay, here channels are closed. Okay, and this is polarized. Polarized membrane. Clear? Done? All of you? Any doubts? Everything is done. Okay, next class we will do synapse, and then we have synapse. After that we have brain. Okay, you have to learn about brain. Clear, students? Yeah, sure. Okay, so yes, thank you so much for joining, and students, you still have a chance to avail the DNA batch. Okay, I will show for students who find it very difficult to join the batch. See, just go to the video that you're watching right now. Right, this is the video that you're watching. Open the video, go down, you can see all the links if you want. And students, see NEET treasure box also. If you want to click on this, click on the NEET treasure box, and you can see all the notes are here, links are here. Okay, I'll update the notes as well. See, almost all my notes are here. Okay, see, all the notes are here. Can you see? Link of all the notes are here. Don't miss this. Every single class, whoever is finding it difficult, be it physics, see, physics is there, chemistry is there, 10 years PYQ is there, NCERT line by line also it is there. See, biological classification. If you click on this, you'll go to the YouTube link. If you click on this, you'll go to my notes. Okay, you just enter your phone number and email, and automatically you'll go to the notes. If you click here, you'll go to my class. Okay, see, Protista and fungi, NCERT. Got it? So this is very, very important. Go and keep a copy of this. See, one student is already here. Keep a copy of this. You will see all the notes here. See, okay, everything is there. NCERT solutions are there. Okay, NCERT, everything you just have to enter your phone number. Once you log in, then everything will be accessible to you. Okay, I hope the session was really helpful. Second part will be coming soon, and also reproductive health also, second part I'll be doing in this week itself. So thank you so much for joining. I hope all of you enjoyed the session. Do not forget to smash the like button and mark your attendance, and also let me know in the comments how the session was, and if you understood action potential like never before with the help of the sibling example, especially on Raksha Bandhan day, then students, I think you should definitely let me know in the comments, because that's the only way I will get to know if you understood or not. So thank you so much for joining. See you all. Tata, bye bye.